Hi friends, welcome you all. Today we're going to start a new unit, that is fermented foods, and in that we're going to see different types of fermented food, their beneficial health aspects, and we'll see a couple of examples of fermentation processes that are involved in preparation of such kind of foods. So here we'll start with introduction. So fermentation, we all know that it is carried out by different kinds of microbes, and the microbes carried out biochemical changes in food, and it get transferred to some different property of food. So traditional fermented food is not only the staple food for most of the developing countries, but also the key healthy food for developed countries. So as the healthy function of these foods are gradually discovered, more and more high throughput biotechnologies are being used to promote old and new industry. So as far as the fermentation process is concerned, so it is so far oldest process, and this process is carried out in pretty much ancient times, and today this process is carried out at industrial scale with modern technology, and it has the area of this process, it get broader. So as a result of microflora manufacturing processes and product healthy functions of these foods were pushed forward, the application and progress of high throughput biotechnologies into traditional fermented food industries like fermented milk products, say for example yogurt, cheese, as well as fermented sausages, fermented vegetables, keema cheese, sauerkraut, then fermented cereals, fermented beans, and gaining momentum. So all these are industrially prepared fermented food products, and various technologies are used for production of these kinds of foods. So given the further promotion by high throughput biotechnologies, the middle and or downstream processes of traditional fermented foods would be optimized, and process of industrialization of local traditional fermented food having many functional factors but not in small quantity would be accelerated. So in case of fermented foods, may be defined as the foods which are processed through activities of microorganism, in which the weight of microorganism in food is usually small. Okay, so these are microbially derived foods, and the foods are solely turned by various biochemical activities of different kinds of microorganism, and the influence of microbial activity on the nature of food, especially in terms of flavor and other organoplastic properties, is profound. So a few fermented foods of economic and nutritional importance are listed in this table. So here these are some of the fermented foods of economic and nutritional importance, and we consume these foods on a daily basis. So in case of wheat, it is fermented to produce bread, okay, and we eat bread on a daily basis. Then milk, these cheese, as well as yogurt are milk based fermentation products. Then in case of maize, so ogi, akmu, kokonte, so these are maize based product. Then cassava, so it is basically related to South African countries, and gari, fofo, akpu, lafun, so these are fermented food products that are made from cassava. Then in case of vegetables, sauerkraut is a popularly fermented food. Then pickles, cucumbers, they are derived from vegetables. Then simulated beverages like coffee, tea, coca, again they are made up by these processes. Then legume and oil seeds, it contains soya sauce, then miso, sufu, then orgam, then idli, ogli, then dawa, then ugba. Then in case of fish, so fish sauces is made. So here these are some examples of raw material, and after their fermentation product, what type of the specific name is given to that fermented product. So these are listed in this particular table. So in case of fermented vegetables, so like the fermentation of other foods, vegetables have been preserved by fermentation from time immemorial by lactic acid bacteria, and a wide range of vegetables and fruit including cabbage, olives, cucumber, onions, peppers, green tomatoes, carrots, okra, celery and cauliflower have been preserved. So only sauerkraut and cucumbers will discuss here. Are there some general principles applied to fermentation of all vegetables and fruits. So in general they are fermented in brine, which eliminates other organism and encourages the lactic acid bacteria. So here we'll study how sauerkraut it get prepared. So sauerkraut is produced by fermentation of cabbage. So basically sauerkraut is derived from vegetable that is known as cabbage, and Brassica oleracea has been known for a long time. So especially selected varieties which are mild flavored are used, and the cabbage is sliced into thin pieces known as slaw and preserved in salt water or brine containing about 2.5% of salt. The slaw must be completely immersed into brine to prevent it from darkening, and kraut fermentation is initiated by Leuconostoc mesenteroids, a heterofermentative lactic acid bacterium. That is, it produces lactic acid as well as acetic acid and CO2 also. That's why it is called as heterofermentative, and it grows over a wide range of pH as well as temperature condition, and CO2 creates anaerobic condition and eliminates organism which might produce enzymes and can cause softening of slaw as well as encourage the growth of lactic acid bacteria. So in case of gram negative coliforms and pseudomonas, soon they get disappear due to acidic condition and give the way to rapid proliferation of other lactic acid bacteria, including Lactobacillus brevis, which is heterofermentative, and homofermentative Lactobacillus plantarum, and sometimes Pediococcus cerevisiae also occurs. So compound which contribute to flavor of sauerkraut begins to appear with increasing growth of lactis, and these components include lactic acid, acetic acid, then ethanol, then volatile compounds such as diacyl acetaldehyde, acetyl isoamyl alcohol, then hexanol, ethyl acetate, then ethyl butyrate and isoamyl acetate. Besides 2.5% of salt, it is important that temperature of about 15% to be 15 degrees Celsius is used. So higher temperature causes deterioration of the kraut. So this is overall regarding preparation of sauerkraut, and it is basically derived from cabbage. Then cucumbers pickling. So cucumber cumin sativus is a scientific name, and it is eaten raw as well as fermentation or pickling. And cucumbers for pickling are best harvested before they mature, and mature cucumbers are too long, ripen easily and are full mature seeds. Cucumbers may be pickled by dry salting or by brine salting, and dry salting is also generally used for cauliflower, peppers, then okra, then carrots, and it consists of adding 10 to 12% of salt to water before cucumbers are placed in tank. So this prevent bruising or other damage to vegetables, and in case of a brine salting is more widely used, and a lower amount of salt is added, between 5 to 8% salt being used, and higher amounts of were previously used to prevent spoilage. So during the primary fermentation lasting 2 to 3 days, most of unwanted bacteria disappear, allowing lactis and yeast to proliferate, and in the final stage after 10 to 14 days, Lactobacillus plantarum and Lactobacillus brevis followed by Pediococcus are the major organism. In case of fermented food from cereals and beans, idli is popular South Indian food that is consumed in the daily at South India, and it is prepared from rice. And idli is a popular fermented breakfast and hospital food which has been eaten in South India for many years, and it is prepared from rice grains and seeds of leguminous mung grain. So it is scientific name that is Phaceolus mungo, or from black gram urad dal, then vigna mungo, which are also known as dal. So this is nothing but mixture of rice as well as dal of different kinds, and the batter is prepared, and that batter is fermented by different kinds of microorganism, especially lactic acid bacteria. Okay, and here the production of idli is given. So it is this is flow chart for production of idli is given. So we'll discuss in this particular process in a separate lecture. Then production of beer. So barley beers can be divided into two broad groups, that is top fermented beers and bottom fermented beers, and this distinction is based on whether the yeast remains at top of the brew or top fermented beers or sediments to bottom fermented beers at the end of fermentation. So raw material for brewing. So beer is mostly derived from barley, then malt, then adjunct, then yeast, the hop and water. So brewer's yeast, so yeast basically used in fermentation of beer, and that yeast is known as brewer's yeast. So yeast in general will produce alcohol from sugars under anaerobic condition, but not all yeast are necessarily suitable for brewing. So brewing yeast, besides producing alcohol, are able to produce form of what sugar as well as proteins in a balanced proportion of yeasters, as well as acids, as well as higher alcohols and ketones, which contribute to peculiar flavor of beer. So here the brewery process is briefly given. So in that, first process is malting, then cleaning and milling of malt, then mashing, then mash operation, and the wort boiling treatment, and fermentation and storage or lagging and packing. So these are different steps of brewery processing is given here. So the malting, so we'll discuss these processes in detail. So here the purpose of malting is to develop amylase and protease in grain, and these enzymes are produced by the germinated barley to enable it to break down carbohydrates and proteins in grain to nourish the germinating seeds before photosynthetic system are developed enough to support the plants. So malting is basically the process to develop amylases as well as proteases in grain. Then cleaning and milling of malt. So the purpose of milling is to expose particles of malt to hydrolytic effects of malt enzymes during the mashing process, and the fine particles greater the extract from that malt. Then mashing. So mashing is a central part of brewing. It determines the nature of wort, hence the nature of nutrient available to yeast, and therefore the type of beer produced. So the purpose of mashing is to extract as much as possible soluble portion of malt and to enzymatically hydrolyze insoluble portions of malt and adjuncts. So the aqueous solution resulting from mashing is known as wort, and the wort is boiled for 1.5 hour in stainless steel kettle. So when corn syrup or sucrose is used as an adjunct, it is added at the beginning of the boiling. So hops are also added, and some before and some at the end of boiling. So hops are dried cone shaped female flower of hope plant, that is humulus eupulus, and the importance of hope in brewing lies in its resins, which provides precursors of the bitter principles in beer, and essential volatile oil which provides hope aroma. So basically hope plant is used for flavor as well as aroma. The purpose of boiling is as follows. So to concentrate the wort, then to sterilize the wort, to inactivate any enzymes, and to extract soluble material from the hope. So these are the boiling purposes of that hope. And the next, to precipitate proteins which forms large flocks because of heat denaturation and complexing with tannins extracted from hops and as well as maltose. Then unprecipitated proteins from hazes in beer, but too little protein leads to poor foam head formation. So these are different objectives of hopping process. Then it is used for removal of volatile acids. So volatile compounds such as fatty acids could be lead to rancidity in which beer is removed, and so these are various aspects of this process. Then fermentation is carried out in a so cooled wort is pumped or allowed to flow by gravity into fermentation tanks, and yeast is inoculated or pitched at a rate of 7.5 to 10:6 yeast per cell per ml. So usually collected from previous brew, and the progress of fermentation is followed by what specific gravity. So yeast is added in fermentation tank, and fermentation process get allowed, then lagering, then packing, and so these are the again post fermentation processes that are carried out in production of beer. So in case of wine, so it is prepared from fruit. So similar way here, grapes, they are used for preparing wines. So first they are crushed, and they are fermented by using yeast like Saccharomyces cerevisiae and other species, and these are several controlling parameters of wine fermentation. So their specific temperature is maintained, as well as oxygen and flavor development, these are monitored at regular intervals. And aging and storage, in case of aging and storage, so wine is stored for longer time for to develop flavor and to develop other characteristics. So these are different processes of wine making. So here this is brief overview of fermented food. In that we have covered several fermented foods as well as beverages. So we'll see other details in next lecture. Thanks.